Okay, good afternoon. Welcome to the Conservation Outcomes webinar series. My name is Liz Marshall, and I am the Outcomes Team Leader for NRCS's Outcomes team. We host this webinar series to highlight activities across NRCS related to its efforts to assess and quantify the impacts associated with its conservation programs, as well as to develop the science necessary to inform effective conservation delivery. If you'd like to be notified of future webinars, you can subscribe to a mailing list at the Conservation Outcomes webinar series website, and there's a link to that site in the box on your screen entitled Today's Links. It's in the lower left of your screen. So we'll get started with today's presentation in just a minute, but first a few logistics. Oops, that's not one. Today's webinar is being recorded. All participants are in listen-only mode. So please type questions and comments into the Q&A box that you will shortly see appearing on your screen. As many questions as possible will be answered at the end of the presentation. If you're having difficulty with audio, computer or mobile device headsets can sometimes help with audio quality and volume. But if you continue to have poor audio or a weak connection and that prevents you from participating today, the webinar recording and slides will be available by Monday at the Conservation Outcomes webinar series webpage, and the link to that site again is on your screen, is in the box on your screen labeled Today's Links. Also, please note that the presenters have provided a number of relevant URLs under the Today's Links box, including some video links that supplement the material that will be presented today. They've also provided a handout of today's slides, which is available to download from the box entitled Today's Handout. And finally, if you'd like to make the slide presentation box larger, you can use the icon with the 4 corner arrows that appears in the upper right of your presentation box to turn on and off the full screen view. I'm now pleased to turn the microphone over to Dr. Louis Tupis. Dr. Tupis is Deputy Chief for Soil Science and Resource Assessment here at NRCS. After the Deputy Chief's introductory remarks, we'll turn the webinar over to Charlie Rewa. Charlie is the NRCS's Wildlife Component Leader for the Conservation Effects Assessment Project, and he will be moderating today's webinar. Thank you, and over to you, Dr. Tupis.
Thank you, Liz. Hello, everyone. Welcome to the latest installment of NRCS's Outcome webinar series. As you know, NRCS has a long history of working with farmers and ranchers to help them conserve the natural heritage and sustainability of their operations. These webinars give us a way to communicate to our stakeholders the importance of voluntary conservation programs and the results of the funding that is provided through the Farm Bill for these efforts. For nearly 20 years, our Conservation Effects Assessment Program has been documenting outcomes of our work on a variety of resource concerns. CEAP works closely with our science partners and other agencies and academia, and outcomes generated help inform improved conservation delivery across the agency. Today's webinar is another great illustration of how science-based CEAP assessments can generate important insight into how planners and producers can improve the land for at-risk wildlife species and overall sustainability. Healthy southwestern streams and associated riparian habitat can tell us a lot about the condition of rangelands and wildlife, and also tell us about the condition of those habitats. Today, we're excited to share a compilation of findings of studies supported by CEAP and other science collaborators in partnership with Northern Arizona University. This is focused on improving our understanding of how the endangered New Mexico meadow jumping mouse, a species intricately tied to the southwestern riparian habitats, responds to various habitat conditions, and how producers can help this at-risk species on productive rangelands of the Southwest. Thank you for joining us today. So I'd like to turn this over now to Charlie Rewa.
Thank you, Louis. Again, I'm Charlie Rewa. I'm the NRCS Wildlife Component Leader for the Conservation Effects Assessment Project, or CEAP. Thanks again, everyone, for joining us this afternoon. We're excited to share some outcomes findings and science generated from nearly 7 years of CEAP-supported studies aimed at improving our understanding of the habitat needs of the endangered New Mexico meadow jumping mouse, and to use that understanding to help inform our work with ranchers and landowners in managing riparian habitats in the Southwest. On the agenda for today's webinar, first, I'll share a few words about the SEEP wildlife component, and then turn it over to Dr. Carol Chambers, who is a professor of wildlife ecology at Northern Arizona University. We should have some time at the end to address some questions that come in during the chat, so please feel free to put those in as we go along. So the SEEP wildlife component, well, SEEP has lots of different components, but today we're just focused on the wildlife component of that. And our objective is to document fish and wildlife outcomes from NRCS conservation work and to use those outcomes to inform more effective conservation delivery. We work across agency divisions and headquarters, our state offices and others to identify the priority assessment needs that we conduct. We rely on cooperative partnerships with the wildlife science community, including universities, NGOs, and state and other federal agencies. So since 2005, SEEP Wildlife has been supporting outcome-based assessments through over 80 different cooperative agreements with our science partners. Assessment findings, reports, and other documents can be found on the SEEP website included in today's links. So the condition of stream systems in riparian areas in the American Southwest has been a concern, an emergent concern in recent decades. So in 2012, the southwestern willow flycatcher was designated a national target species for a Working Lands for Wildlife effort. And while the flycatcher is the flagship of our work with landowners to restore riparian areas in the arid Southwest, there are 84 other species tied to these riparian habitats that are also covered. The endangered New Mexico meadow jumping mouse is one of those species. And until recently, little was known about its habits and its habitat needs. So in response to the increased attention to southwestern riparian conservation and requests we received from state and regional NRCS contacts for increased knowledge of this species' needs, SEEP Wildlife has been supporting a number of assessments to help us understand how the New Mexico meadow jumping mice respond to conservation work and to inform more effective delivery for conservation for that species. So since 2015, we've been working in partnership with Dr. Carol Chambers at Northern Arizona University and other state and federal science partners to assess the habitat needs of this cryptic species and to help us understand how conservation practices benefit it and how we can help producers support healthy riparian systems for jumping mice and the other species associated with these working landscapes. So as Louis mentioned, today's presentation is a compilation of findings from this science partnership and highlights some of the outcomes and science products developed through this body of work. So with that, I'd like to turn the floor over to Dr. Carol Chambers for the main webinar presentation, and I'll be back at the end of the webinar for the Q&A session. So Dr. Chambers, the mic is yours. Thank you.
Thank you, Charlie. Can you hear me okay?
Yes, we can.
Okay, great. Let me get, oops, there we go. Well, thank you for the opportunity to talk about the species and what it tells us about riparian management. I study habitat relationships, and I like working with agency personnel to help solve management questions. So working with NRCS, a big question has been to understand the habitat that these animals use. I'm going to talk about the work we've been doing on this species and how it's informed management approaches, restoration, and development of a wildlife habitat evaluation guide. And I love questions. If you have any along the way, please drop them in the chat, and I'll look forward to addressing those later. To give you a perspective on how unusual this animal is, I put together a couple of quiz questions. So how far can a jumping mouse travel in a short period of time? And how far can a human jump if we could jump as far as a jumping mouse? So you can pick your answer here. And I'm going to move straight into the answer. So see if you got the right answer. How far can a mouse move, a jumping mouse travel in 25 minutes? 1,640 feet. We tracked a female returning upstream to her nest one night. She moved 1,640 feet in 25 minutes, so about 66 feet per minute. And half the time she was moving upstream. These guys are very good swimmers, so this is quite a challenge for a little animal moving. So I hope you're as impressed as I was. And second, how far could I jump? I think this is a good gee whiz factor. If it was me, I would bounce 27 feet in one leap. So these guys have quite the capacity to move when they want. I also want to ask you a couple more questions. I don't expect you to know the answers to these right now, but I do think you will know these by the end of this talk. These are a couple questions that relate to habitat and diet. They're aspects of the jumping mice that we didn't know prior to our studies. So see what you think. These are just true or false questions. Sedges and rushes are the main diet for jumping mice in the Southwest, and introduced plant species have displaced most native species in New Mexico meadow jumping mouse habitat. True or false? And I will come back to these in a little bit. I really appreciate the funding and support over the last 7 years that we've been working on the species. NRCS has been a great help in our understanding of the species and management knowledge that we have now. And I also want to acknowledge the collaborators, the grad students, the genetics and field technicians who did most of the work. And I'd also like to recognize the photographers who supplied most of the photos for today that you're going to see in this talk. I like to use a lot of visuals. We work in some beautiful places, so hopefully you'll enjoy your visit to our field sites. And for perspective, all the people listed on the previous slide helped with setting traps or track plates. They handled small mammals, they collected samples for analysis, they surveyed vegetation at sites, they analyzed samples in our labs. We created a database of locations for mapping the species. We collared a lot of jumping mice and stayed up a lot of nights tracking them. So there's a lot of sweat in this project. I also like to remind people that we work in the Southwest, but we are not desert and cacti, as many people think the Southwest represents. Our work occurs at elevations from about 4,500 to 9,500 feet, and we're working in forest and forest meadows like you see in the picture here. Because we're in an arid system, riparian systems are incredibly important, but they represent less than 2% of the land area in the Southwest. They might be uncommon, but functionally they're really important because they help control water pollution, they reduce erosion, and they mitigate flooding. This is one of our high-quality sites for jumping mice that flooded last year during a very heavy rainstorm. And you can see there's not a lot of erosion because of the well-connected floodplain that disperses water and the vegetation that reduces erosion. Another important function of riparian areas is wildlife habitat. 80% of all vertebrates and 70% of threatened or endangered species rely on or spend some portion of their life cycle in these riparian areas. And this is the New Mexico meadow jumping mouse, an endangered species perched on a rock in a riparian area. The species was listed in 2014 as endangered, and at the time we didn't know a whole lot about its habitat use, its movement capabilities, or hibernation sites. And because it's listed, we're now studying this rodent, but it's also telling us a lot about riparian function and what we need to do to maintain habitat and have good functional riparian habitat. So threats that led to listing included habitat degradation or loss. Primary threats ranged from overgrazing to water use practices to recreation. And we knew something about other jumping mice that occur further north in the US and Canada, certainly in more mesic sites further north. But we didn't know a lot about the ecology of this particular species. We suspected it was a riparian obligate, something that requires riparian areas for its survival. We thought it to be nocturnal and hard to capture, but there was a lot more we needed to know to be able to effectively manage the species. I started studying the species in 2015, just after it was listed. There were a lot of questions that needed answers to address recovery, and these are some of the questions that I and my students have taken on over the years. And if you read down through this list, you might notice that some of them are at smaller scales than others. We kind of started at the small scale, like can we actually capture these animals? How do we detect them? What do they eat? Where do they live? And then as we learned more, we were able to scale up and look at movements and dispersal. And to date, we have been able to cover 10 of these questions. We're currently working on this last one. So we're making a lot of progress, and we're getting a much clearer picture of what a functional stream should look like for the New Mexico meadow jumping mouse. So this is the answer. This is what your stream system should look like for jumping mouse. This is the West Fork of the Black River in northeastern Arizona. It's a geomorphic reference site for the jumping mouse, and it illustrates the most desirable physical condition for a natural, stable system with excellent floodplain connectivity. So how do we determine this is prime functional habitat? I'm going to come back to this picture at the end of the talk, and I hope you'll be able to identify elements of the habitat and stream condition that support the jumping mouse at that time. Working with NRCS and our data, we've produced a draft wildlife habitat evaluation guide for the New Mexico meadow jumping mouse. You don't need to read the detail here, just to know that we have prepared this draft. We'll be field testing it soon. It describes 12 elements that are important to stream quality and jumping mouse survival. And I'm going to come back to this WEG periodically during the talk and at the end just to show you how we drew these conclusions and put them in the WEG. Okay, I'm going to start you off with a little bit of jumping mouse ecology. Jumping mice are not mice. They are actually in their own taxonomic family, the Zapodidae, so they're not with wild mice that are in a different family, the Muridae. They are mouse-like, but they're also kangaroo-like. They have a very long tail, which they use for balance, whether they're hopping, swimming, or climbing. They can leap very long distances when alarmed, and they are nocturnal and solitary. They've mastered a variety of locomotion styles to help obtain resources in their environment and to avoid danger. This animal is leaping. It's becoming a very aerodynamic little missile. But these animals are also excellent climbers and swimmers. They can move stealthily through undergrowth. I like to compare them to basketball players because of their ability to go up very easily. And this is kind of unusual in the small mammal world. If you want to see these animals on the move, you can look at our YouTube link and see a short clip of some of the video we've taken in the field when we've been able to see these animals. So their morphology and modes of locomotion are unique, but there are other unique aspects about the jumping mouse. This animal is on a scale, it's not dead. It's in torpor. Jumping mice hibernate, and they also go into short torpor bouts when needed. They hibernate 7 to 9 months out of the year. So why would an animal, small mammal, hibernate? Generally, it's because the lack of resources needed for survival. And I think these reasons will become clear as I tell you more information about their diet. So they have a very short active period of just 3 to 5 months in which they have to accomplish everything they have to do. They have to reproduce, raise young, and then gain enough fat to survive the next 7 to 9 month hibernation period. They appear to have low reproduction rates, maybe just 1 litter of pups per year. So when their populations are down, it's going to take them longer to recover those populations. Okay, this is a fat jumping mouse, and that's a good thing. Animals in the summer, during summer, when they're just moving around, traveling, reproducing, weigh about 20 grams, but in the fall or in August, September, just before they go into hibernation, they sometimes up to double their mass. So they go from 20 to the largest one we captured was 39 grams. So in general, they have to add an additional 6 grams of mass or they die during hibernation. And this is, so it's really important for these animals to have a high quality diet just prior to going into hibernation. So they need to sit and stuff themselves and not move very far to acquire the fat resources that are important. Okay, so what does it take to survive? We need to know a bunch of these things: species distribution, habitat, diet, home range, and so on. And I'm going to cover aspects of studies we have conducted to help you understand the WEG that we've developed to predict jumping mouse, or to look at habitat for jumping mice. Okay, do we know where jumping mice occur? They've been documented in Colorado, New Mexico, and Arizona. This map shows major rivers in blue and the current distribution of New Mexico meadow jumping mouse in orange by county. So it was generated using records from past work. And it's a rather coarse look at distribution. So how do we refine a map like this to better predict where jumping mice are going to occur? What we've done is look at the species distribution using a modeling approach. Jose Martinez has led the development of this model, so we used all past records that we could acquire and our capture data from the past 7 years to generate over 2,000 locations for jumping mice. And we spatially rarefied these occurrences to account for spatial autocorrelation. We ended up with 585 locations for modeling and used some pseudo-absence locations for comparison. We used Maxent software to model the species at a fairly small grain, 30 by 30 meters. And then we're modeling across the range of the jumping mouse. So you can see the variables modeled included vegetation, distance from stream, and bioclimate variables. And the ones that were best predictors in the model were distance from stream and a few of the bioclimate variables that included temperature and precipitation, and not so much the vegetation, the National Vegetation Classification information. That really wasn't as important here. So if you look at the map, the blue area represents the area that was modeled, and the red areas, the color ramp goes from red to blue, with red being the highest likelihood of occurrence of the jumping mouse. So hotter areas are more likely to have jumping mice. And I just want to blow this map up and show you how we can apply this. If we look at this area in the circle, that's southern or sort of eastern Arizona, blow that up, look at the map on the left. You can see the red lines following riparian systems. So we can use the detail in this map to find areas that we haven't surveyed before and then go in and see if we can detect new populations of the animals. So we know better where to look, but what are we looking for? What type of habitat is optimal for the jumping mouse? We focused on riparian areas since jumping mice are riparian obligates. I mentioned vegetation wasn't a strong predictor. And if you look, this is one example of our riparian area, and I want to show you another. This is high elevation, and this is lower elevation. So you see it's quite variable in the vegetation that occurs in these areas. So the jumping mouse isn't keen so much on vegetation as it is on other aspects. So we sampled riparian areas known to have jumping mice and compared them to areas without jumping mice at 2 scales. First within Arizona in the White Mountains, where the star is on this map, and then second throughout the range of the species. We sampled mostly on national forests because we knew the species was present. We had access to a lot of sites, but we also sampled some private and state lands whenever possible. In Arizona, we sampled 75 sites. We used a couple of methods. And a common method is to live trap small mammals with Sherman live traps. You need a lot of people on site. It's very time consuming, and you have to be able to move quickly among sites. And to cover so many sites, we had to come up with another approach to detecting jumping mice. And this is Rachel Harrow, an undergraduate at the time, who led the effort to, in the field to develop a non-invasive approach to detecting jumping mice. So we found that we could use track plating. The animals have really unusual footprints. The jumping mouse on the left is giving you an example of hind feet that's very elongated feet and toes, and the forefeet have very elongated toes. And they're really different from the other animals that occur in the same area, voles and deer mice. So we developed this inexpensive track plate. We just used plastic boxes and some carpenter's chalk and oil and an ink pad. The animals come into the trap, explore, eat the bait, and leave a footprint. So this helped us accomplish a 75-site survey during 2016 in a 2-month period. So we were able to select sites using specific criteria and looking at specific elevation ranges and vegetation types, and then making sure we could move among sites and either collect data from live trapping or track plating. So we also sampled a range of sites that included livestock raising and recreational use, and some sites, this is a site that had actually been restored over time on private land. And along with small mammal tracking or small mammal trapping or track plating, we also collected vegetation and habitat data at all these sites. This is what we found. We found 8 variables that had the greatest predictive power. So the taller the vegetation, the better for jumping mice. The wider the stream, the better. The more alder, the better, and so on. And I want to take a couple of these variables and show you how they actually model out. So I want to take mean total vegetation height and percent alder. So when you look across this graph, the mean vegetation height is on the x-axis. So we're going from 0 to 40 inches, and we're looking at the occupancy estimate of basically 0 to 100% or 0 to 1 on the y-axis. So you'll see that at about 24 inches of vegetation height, half the sites are likely, or the site is about 50% likely to be occupied by jumping mice, but the taller the vegetation, the better for jumping mouse. And with alder, even a little bit of shrub cover was likely to dramatically increase the presence of jumping mice. So in this case, we're looking at maybe 4% alder cover along an 800 to 1,000 foot length of stream to get 50% occupancy. So again, a little bit of shrub cover goes a long way. And we looked at alder cover because that was the dominant shrub in the Apache grazed area where we were working, the White Mountains. I mentioned that we had some sites that were grazed by livestock in the study. We found jumping mice in areas with or without livestock grazing. So this variable was not a predictor of occupancy. And we think vegetation height is the proxy for grazing. So that was a better measure of the amount of grazing than just presence or absence of grazing. So this summarizes our habitat model for Arizona. Jumping mice liked sites with tall vegetation, wide streams, some sedge, forb, and alder cover, moist soils. And I want to quickly show you this comparison across the region. We wanted to look region-wide and see if these same variables held up. But we also added plant species composition into our model. So we hired the same botanist to survey all of our sites for plant species and categorize their dominance across all sites. So the results of our regional model were similar to the Arizona model, but they included plant species richness as a predictor. So if you look, you'll see that total vegetation height, sedge, alder, and forb cover are also in this model, as well as stream width. So very similar results. And something that I found really important and interesting was the fact that most of the riparian plants were native species. So on the x-axis is a site. Each bar represents one site. And on the y-axis is the number of plant species. In green, the native plants, and on top of that, the number of introduced plants. So you'll notice the large amount of green. So most of the plants at these sites were actually native plants. Even though they might not be very tall, they were still hanging on to the site despite whatever disturbances occurred. And this is across the range. So 2 locations in New Mexico and 1 in Arizona. So based on our regional model, we are seeing very similar things, adding in plant species diversity. But again, this really tells us what jumping mice need. And so across the range of the species, we know we need tall, dense vegetation, wide streams, good floodplains, alder, forb, and sedge cover, and plant species richness is also important. Okay, so in terms of applications from this work within our WEG, there are, 7 factors that inform the 12-element WEG. And we can also use the species distribution model to look for new areas where jumping mice occur, but we just haven't sampled there yet. So next we wanted to look at diet of the animals. This was mostly unknown because it's really hard to observe these animals in the field and see what they're feeding on. And if you want to look at their gut contents, you have to kill them. And so for an endangered species, that's not a good approach. So we looked at stable isotopes, shotgun metagenomics approaches, and metabarcoding. So I'm not going to go into detail on how stable isotopes work, but they can tell you whether the diet of the animal is C3 or C4 or a combination, and they can tell you whether animals are herbivorous or carnivorous or in between. And in this graph, the circles, squares, and triangles represent our jumping mice, individual jumping mice, and the X's are plants. And you can see the jumping mice tend to be C3 feeders, and they're largely herbivorous, although there are a few that are feeding more on C4 plants. So this gave us an idea that they're really focused on plants, and with our shotgun metagenomics approach, this basically tells you information on anything that can be genetically identified in the sample. So our results, to me, were really surprising because they showed a real diversity of plant families, and I was looking at what species might occur in the region where we were collecting these data. And this is a list of potential species. So in the, let's see, a good example that surprised me, well, the Geraniaceae, a wild geranium occurs commonly in this area. So we're starting to understand that jumping mice feed on a salad bar. And with this knowledge, Dan Sanchez took on identification diet for his dissertation. He used metabarcoding. So over a 5-year period, we captured 145 individuals for the study. And we have a great approach for doing this work, and that is through a relatively non-invasive method of picking poop from traps. So this is a very happy technician who is pulling fecal samples from a trap that had a jumping mouse in it. And there's a ton of information you can get genetically from a sample like this. So I'm not going to go into details here, but Dan genetically identified plants, arthropods, and even looked at for hypogeous fungi in each individual's diet. And he found some really cool stuff. He found that forbs and graminoids were found in 95% of the individuals. He detected up to 9 plants per individual, so again, getting back to the salad bar approach. He also identified arthropods in 81% of individuals, and most of these arthropods were Lepidoptera. He did find hypogeous fungi, but only in a few individuals, and those were mostly in August or September, probably when our monsoons were happening and we had a moisture condition. So this is a little bit more detail on the plants that he looked at, since they seem to be the dominant portion of the diet. Dan identified 117 families, 161 genera, and 84 species of plants, arthropods, and hypogeous fungi. So in this chart, we're just looking at plants. Each box represents a species of plant or a genus identified as a diet item in a jumping mouse. The size of the box represents the number of jumping mice that consumed it. So the larger boxes are plants that were selected more by jumping mice. So if you look at this, what looks like the most dominant set of plants, look for the largest boxes, and you should see that geranium and rudbeckia and geum were some of the more common forbs, and some of the more common graminoids included poa species and triticum species. So these animals have a really wide dietary breadth. They feed on a variety of plants, and they also feed on insects. Just to give you an idea from a mouse's view of what a salad bar should look like, this is ideal late summer habitat. These plants are the foundation of the diet for these animals. I think the arthropods are more incidental or maybe opportunistic. They might be important early in the season, just after emergence from hibernation. But using these genetic approaches that were pretty non-invasive, we have looked at the diet of the species across its range for a whole bunch of individuals, and so we feel like we have a very strong understanding of what the animal eats. And the cool thing is you can apply this to other species. We've done this with pronghorn and mule deer as well now too. So also positive is the large percentage of native plants that we discovered in riparian sites and in the diet of the jumping mice. These are pictures of the same area at slightly different vantage points and 2 different years. In the picture on the left, the area was grazed by livestock and it was used for recreation. I was told that vehicles would be driving back and forth across the river here. In 2014, it was fenced after it was designated as critical habitat for the jumping mouse. And so just 2 years later, I was there surveying for jumping mice, and I took the picture on the right. I'm a little farther away from the stream, but where the tall vegetation is in the sort of foreground is where that stream would be. And you can see how quickly the area had regrown. You're seeing tall vegetation, a variety of vegetation. And so this plant community very quickly recovered from disturbance, and it was supporting. We captured jumping mice and continually captured jumping mice at this site. So in terms of applications, we've added 3 factors to the WEG that are derived from the information I was just talking about. And we also were asked for our species list so that it could be used to develop a seed mix for more rapid restoration of streams. Okay, up next for us was home range. We had learned how to capture and detect animals. We were well underway to understanding habitat and diet, but we didn't know how far the animals could move, and we didn't know if they were always in riparian areas or if they moved upland, if they could disperse across uplands. So we looked at this using 2 approaches, home range study and genetic applications. And I'm going to start with home range first. There was one previous study. This was conducted at lower elevation. So we did this work across the range of the species for 4 years and used live trapping and radio collaring, and we were working at mostly these higher elevations. So we set out trapping transects along streams. We captured small mammals. We had to learn how to radio collar small anxious animals, which made me anxious as a larger animal. And it also gets harder as your vision gets worse. But one person basically holds the mouse and tries to keep it calm while the other person slips the collar over the head and tightens it. And then once the collar is fitted and checked, the animal is released. And then because these are very small radio collars, we have to physically track individuals. We can't use GIS or GPS telemetry for these guys. So we're out in the field tracking a minimum of 7 days with 30 independent fixes, but sometimes we were working for weeks on end and all night. And then during the day, we would also look for activity to see where the animals were. When we found the animal, we would mark the location, like this location in a patch of thistle and stinging nettle. We discovered later they actually eat stinging nettle and sit happily up on them, so I envy them that. And we discovered that during the day they use small bolus nests that are about fist size. They're well concealed under cover of tall grasses or under grass mats. And this really helps with thermal cover and predation protection. So on the picture on the upper left, you might be able to see a hint of that nest that the technician is pointing to. And if you look on the right, try to find the nest on the right. In the next picture, I'm going to show you where it is. So there it is. It's not so easy to see. They're very well concealed. And again, they need a lot of cover to protect themselves both from temperature extremes and predation. So we worked to get 70 home ranges across the range. We collared well over 100 animals. Some animals were predated, some disappeared, but we were able to, with 70 of these animals, generate 2 kinds of home range estimates, minimum convex polygon and kernel density. And I want to show you an example of one of our home ranges depicted both ways. So this is a minimum convex polygon for a jumping mouse we tracked in 2019. The purple area is an exclosure with an 8-foot tall fence to exclude elk and livestock. The red outlines the minimum convex polygon. The blue lines represent the stream, so the riparian areas through this area. The blue circles are our nocturnal locations for the jumping mouse, this individual, and the yellow are day locations. So if you draw a boundary around the outermost points, you've created the minimum convex polygon area. And if you notice, the yellow dots sometimes have numbers. These yellow dots are day nests, and the number is the number of times a nest was used so that you can see there were 6 day nests if you count them. 3 were used repeatedly in this home range, and all were within the area protected by the exclosure. This is the same location as a kernel density estimate. It's usually considered, this kernel density approach is usually considered a more accurate measure of home range because it excludes areas that the animal never used. So to summarize everything from our home range study, we found that jumping mice use about 3 hectare home range. That's about 7 acres. It's larger than those that were documented in the previous study. We also got movement data, so we can know, how far can the animal move since its last location. That can give us an idea of how it might disperse when the animals need to move to disperse. So 273 meters is about 900 feet. So these animals can move quite long distances in short periods of time. And the other number provided gives you an idea how far on average jumping mice move away from streams. So 11 meters or about 35 feet. So they're really not going that far from streams. And to emphasize that, this is a home range, another home range. Notice the blue line indicating the stream, and that the yellow and blue dots occur pretty much on the stream. So we found through telemetry that non-riparian travel doesn't happen much. We didn't actually document it. So riparian function is really critical to the success of this animal. So we also looked at this question genetically. Do jumping mice travel just along riparian corridors, or can they move cross-country in less hospitable habitat like a ponderosa pine forest? And can they move among watersheds? This is more work by Dan Sanchez. He did the diet work on jumping mice, and he also looked at population structure, again using fecal DNA mostly. He looked at nuclear and mitochondrial DNA across 3 watersheds in Arizona with a fairly large sample size of 147. So he's looking at gene flow. So this was his question: do disconnected watersheds structure populations? He looked at these 3 watersheds: the Little Colorado River, the LCR, the San Francisco River, SF, and the Black River, BR. And you can see areas where we sampled in dots. And he was he was asking 3 different questions or hypotheses developed on gene flow. Was there no gene flow at all between watersheds? Was there occasional gene flow, or was there a lot of gene flow? And I also want to note that we do have monsoon rains, usually July to September, so later in the summer, that can make upland areas that might generally be inhospitable to a riparian animal, that can make it a little more hospitable. But the headwaters for these locations, for these 3 watersheds, are 2 to 6 kilometers apart. So that's quite a distance to move between areas. So this is what he found. He found that occasional gene flow hypothesis appears to occur here. So there was discernible structure for 3 populations, 1 per watershed, but there was some movement between watersheds, and what he found was some ability to move, or some movements from the LCR, the Little Colorado River, into the San Francisco River, and some movements from the Black River into the San Francisco River. So although animals aren't moving freely among all 3 watersheds, this isn't one big jumping mouse population, there does appear to be some movement going on. And he did find that on the eastern side of the Little Colorado River, the area in the box, that there was some disruption of habitat that was potentially affecting gene flow. There was more housing development, more livestock grazing, and water diversion. And so the gene flow was only occurring one direction, from the LCR into the San Francisco River. And then a lot of the land from the San Francisco River to the border with New Mexico is privately owned. And so there's some great potential for increasing movements, increasing connections among these watersheds. So Dan's answer was yes, disconnected watersheds impede movement, and they genetically isolate jumping mice, but it looks like we can help reduce that impediment, I'm not sure that's a word, but it looks like we can do some things within watersheds to increase gene flow. So how did this inform our work? Telemetry and DNA let us know what we should add into this WEG, and it also provided information that will relate to captive breeding and translocations of jumping mice in the future. So both of these are options for recovery of the species. So I want to end with some new information that Charlotte Rosansky, working with NRCS, just completed. She studied the fluviogeomorphology of streams to understand what conditions are associated with presence of the jumping mouse. And she wanted to understand the physical cause and effect of degradation in fluvial systems to apply this to watershed restoration. This was her thesis question, and she worked with NRCS staff to extend their pilot study that started with data collected for 20 stream sites. And at the time, they also identified this as a reference site for jumping mouse. This is that Black River site that I mentioned initially. So these reference sites can help guide restoration. So recall that streams in the Southwest are really important, but they vary in their degree of degradation. The stream had a railroad along it at one point, and it's had livestock grazing for decades. And so we're looking at areas that have different levels of need. Here's another example of a stream in a degraded state. The stream isn't well connected to its floodplain anymore. So Charlotte worked in Arizona. Since we had a lot of information in our largest sample size here, she looked at the sites on 3 watersheds in the White Mountains, and she compared sites with or without presence of jumping mice. And these are the data she collected, a number of variables, and in her analysis. So I'll point out a few of these that were important and that we're building into the WEG in the next few slides. What predicted use by jumping mice? A lower bank height ratio was a very good predictor, higher sinuosity, and channel evolution stages. And in working on the WEG, we realized that bank height ratios are not easy to measure in the field, not quick to measure. So we're focusing on sinuosity and channel evolution stages, and that's what I'll tell you about just in the next slide or so. So sinuosity was a strong predictor of the presence of jumping mice. It characterizes the channel pattern. We did not find them on streams that were straight channels, but we did find them on these channels that were more that were still well connected to the floodplains, so very sinuous. And then in terms of stream evolution, these stream evolution models, this also predicted presence of the jumping mouse. So this is a simple depiction of how a stream might change over time if it's disturbed. And we found that jumping mice were using stages 4 and 5, so these are widening and recovering stream systems. We didn't find very many stage 1 stable stream systems in the Southwest, but jumping mice were not on stage 2 and 3. So in terms of application, we're using this information to feed into the WEG and also developing recommendations for restoration. So what are these recommendations for restoration? So basically, let's see what systems are degraded, which are no longer functioning, and target those for restoration. We consider a 600-foot stretch of stream that is low-quality habitat to be fine for restoration, because this can be a barrier for jumping mice to move across it. Fencing has been very useful to exclude some or all grazers and prevent overgrazing. Beaver dam analogs can reduce flow. They can help rebuild streams. If you look at the high bank on the back of this picture, or towards the back of this picture, you can see where how deep the stream channel has gone. There were a number of beaver dam analogs placed along the stream, and then a beaver came in and altered a few. So these have been successful in helping slow stream movements and reestablish vegetation. Last year, we encountered cattle incursions at some sites that we surveyed. Grazing was generally light and short duration with few cattle. So some level of grazing might be compatible with maintaining habitat for jumping mice. But we need metrics to compare grazing and utilization to presence of jumping mice. And so right now, I think our best approach to measuring grazing effects is looking at vegetation height and density, and those are what we've built in our WEG. So this is a success story on private lands. The owner excluded elk by putting up an 8-foot fence. He continuously grazed just a few livestock. He planted shrubs. He removed invasive plants. He even got beaver back into the system. And you can see the original channel, how much higher above. If you look in the background there, you can see how that the stream was really down cut. So this stream has really gone back to a channel evolution stage 5 now. So it's gone through a lot to recover here. We did capture jumping mice here, and he achieved this over like a 10, 15-year period, maybe shorter than that, but I know he was working in this area just kind of on his own over that period of time. So we were pretty excited. We caught jumping mice here. We caught animals that were reproducing, so this looked great. But the sad news is that upstream there is now a water diversion that's reduced water flow. We sadly found dead beaver. There isn't much more than a trickle of water running through here. So jumping mice are literally losing ground here. And this property connects areas with heavy elk grazing on adjacent Forest Service lands, and it also connects private lands. So losing this site loses a potential connective space for jumping mice. So this is what I wanted to come back to, and hopefully now based on our findings, you see what this stream shows as good habitat for jumping mice. And the riparian vegetation is really important, but we definitely have to address whether riparian systems are hydrologically and geomorphologically stable. If they've shifted away from a stable state, we have to look at how to return them to that stable state so that they can maintain the vegetation, the riparian vegetation that jumping mice need. There's more work to be done. Jen Zaraka in the upper right is working on, she's using fecal material again. We use a lot of feces on this project to look at breeding cycles so we can get a better idea of population recovery. If there's just 1 litter per year versus 2, that could tell us quite a bit. And Faith Walker on the bottom right is helping describe an eDNA method for sampling. And what we found is we can detect jumping mice from the plants that they're moving on. So we've developed an assay. This is currently in review. And we're also planning to test relocation or translocations of jumping mice to see if we can restore sites, move the jumping mice out, restore the site, move them back in, and how successful they will be. So I want to take you back to these 12 factors that are included in our draft WEG. These will help with stream assessments for jumping mice. And if we have time, I'll let you look at these, think about whether you can now answer these questions or not. And hopefully you got that false. Sedges and rushes aren't the main diet. Forbs and grasses are. And false, that on average most of the plants in jumping mouse habitat are native. So as a summary, we need healthy riparian areas, tall diverse vegetation, and we need to make better connections between habitat patches. So this is a melanistic jumping mouse that we captured. This is not the common color for the mouse, but kind of neat to see. And at this point, I'm not sure if there's time for questions, so I'll turn things back over to Charlie and answer any if there is time.
Okay, well, thanks, Carol. That was a whirlwind of a lot of information. I really appreciate you going through it. There were a couple of questions that came in, and if there's others in the next couple of minutes, I'll try to track them. But this question here is, have you found a strong correlation between species presence and fenced riparian exclosures?
We certainly go back to the same areas for trapping animals, and they tend to have fences. If we're trying to reliably capture animals, fenced areas do seem to have a better quality habitat and more likelihood of capturing animals. I haven't tested that in a statistical manner, though.
Okay. There's another one here about the elevation that says, how might the mouse in these higher elevation sites compare with mice in lower elevation, like the Middle Rio Grande Valley? Would home range be significantly different?
Yeah, that's a really great question. I mean, so at higher elevation, you have a shorter time period for the animal to be active, so it's going to have to be below ground longer than at lower elevation. So you might need really high quality resources for that short period of time for the animal to survive. At lower elevation, though, the vegetation is different, and So the amount of vegetation may be lower. The only thing I can compare to is that that study at Bosque del Apache that I mentioned at lower elevation, they found smaller home ranges, but they're also looking at pretty limited habitat. So unless we could get into other areas at lower elevation, it's hard to predict if they'd be smaller or larger. But certainly the animals are more constricted time-wise at higher elevation. They can't be active as long as at lower elevation.
Okay. There's another question about grazing. It seems possible that the jumping mouse might be somewhat grazing dependent with their habitat needs. Would these areas become more forested if grazing was completely removed?
Yeah, that's another interesting question because it depends on the soil type. I would expect some soil types don't support tree growth well, and so they're going to remain like wet meadows or meadow systems. So I think it would depend on the location. So, you know, if you're in an area that could grow trees, that in the Southwest we might have had repeated fire that would decrease the tree density. So I think, again, it would depend on the specific location. I'm just looking at this question again to see if I've answered it. Would they be, if you removed grazing, would the areas become more forested essentially? And so I suspect that's possible in some areas. I think in a lot of areas that would not be the case, but it would depend on the soil type and the fire regime.
Okay, there's a question here about where do they hibernate? Near the stream?
God, that's another fun question. I didn't show you the hibernation sites. These guys are really variable in what they eat, and they're also really variable in where they hibernate and where they put their day nests. So we've got a total of 10 hibernacula that Jen Zaratka has right now, but we only know details on 3 of them because 7 of them the animals are hibernating in them right now. So we've dug up a couple when the animals were gone, and one was 5 inches deep, one was 13 inches deep, and those were between, I think it was 5 meters and 100 meters from the stream. So one was in a kind of an open meadow, north facing, and the other was in a bunch of riparian, slightly upland vegetation. So it's really hard to classify what their hibernacula look like. We're just getting an understanding of that.
Okay. There's a question about, have you surveyed similar riparian areas in the Gila National Forest?
We have, we did track plating over in the Gila several years in a row, and there have been jumping mice that were detected just across the border from Arizona into New Mexico. We did not detect any at the sites where we sampled. In some cases, they have a lot of elk there, and there wasn't much vegetation in the, it kind of looked like a lawn that had been mowed. So I'm not sure what the potential would be if there was capability to grow taller vegetation there. That might work well.
Okay, I think we have maybe time for one more. Somebody suggested, Carol, you might discuss the Lincoln National Forest situation. Do you want to get into that or no?
Boy, that's complicated. So the Lincoln, I'll try and do this briefly. The Lincoln National Forest has, as far as we know, just one location remaining of quite a few, of at least 10 or 15 that I guess used to be known locations on streams. So as far as I know, there's just one location left where the jumping mouse remains. And the Lincoln is working to expand fencing in that area and try to provide better habitat along that stream system. But the species, the subspecies there, or the animals there are going to be a distinct population like they are in each watershed. So it gets tricky. How do you conserve individuals from that location? And that's where Fish and Wildlife Service has talked about a potential captive breeding project where you take animals from that system and try to recover them using the same genetic stocks that came from that area. But yeah, that's a difficult area.
There's a lot there, and we didn't get to talk a whole lot about maybe how private lands link these public lands, but there's there's a lot we can do to help this species, and we're certainly grateful for the science that you have contributed to help us do the best we can. So with that, I'd like to turn it back over to Liz to close out the webinar since we are at the top of the hour. So go ahead, Liz.
Great, thanks, Charlie. I just want to thank both Charlie and Carol Chambers for today's presentation. Thanks also to everyone who attended today's webinar, and we hope to see you again for future webinars. Thanks and have a great afternoon.